Hello, everyone. Welcome to SKC Science and Technology Webinar Series. I'm Deep Midi. I'm the organizer of this webinar series. This is open to everyone who wants to listen. And we do it in what I call is a fireside chat form. So, and today it's my pleasure to have Professor Giri Narasimhan of Florida International University as the speaker. He's going to talk about something I don't know much about, so this is going to be exciting for me to learn also. He's going to talk about not the social network of people, but social network of microbes in microbiomes. So it's an interesting topic. Why it is interesting? It brings in together aspect of biology as well as computer science. And so this is why this is very, very interesting and important for us. To tell you a little bit more about Giri is that he is known to me for a very, very long time. I met Giri more than 35 years ago when both of us were graduate students in computer science at University of Wisconsin-Madison. So I know him for a very long time. He has his undergraduate degree from IIT Mumbai in electrical engineering, and then he switched to computer science. He has been doing phenomenal amount of work on this area of interdisciplinary area of biology and computer science. And he also has a very broader interest he does for CS education for K to 12 level activities he does. So with that, I will hand it off to Giri. Take it away.
Thank you. Thank you, Deep. Let me share the screen. Give me just a second. Well, thank you for the kind introduction, Deep. Yes, we've known each other for many years, for many decades, in fact. And back when we were young and idealistic. So a lot of things have happened since then. And today I want to talk about some of the time I've spent thinking about. It says sharing is paused. There you go. Can everyone see this?
Yep, now it is
okay, good.
Okay, great. Go ahead.
So today's talk is titled The Social Network of Microbiomes, and the title will slowly become clear. What I want to tell you about is that the topic is relevant for the day because we live in these times that are very strange. We are in the midst of a pandemic caused by a microbe, caused by a virus. And this has struck fear in the hearts of people. And it has dictated in many ways, how we lead our lives. It has literally changed our lives forever. And we think of this as an invisible enemy. It's anywhere and everywhere. We go wash our hands all the time. We change our clothes when we come back from outside. We wash our vegetables more now. When we exchange money, we are more careful. And we are afraid of the air we breathe because we are told the virus can enter us through the nose. So we are constantly bombarded by these scary terms like pathogens and infections and things of that sort. So part of this is relevant because microbes are everywhere and are currently in our consciousness. So that's the backdrop against which we will be talking. This slide shows that microbiomes are, even before this pandemic, they were extremely popular. They were part of the popular press, popular media. Here are 12 books that I just picked from the internet. Shows that a lot more people have been thinking about. What's important to understand here is that is that many people believe, and part of it is, of course, also true, that there is a lot of hype about this thing called the microbiome. Very briefly, the microbiome is a community of bacteria that lives in specific environmental niches. In particular, it lives in the human body, it lives on our skin, it lives in our mouth, it lives in our gut. And then it's also out in the environment, it's out on the desk, it's on the pen that you use to write. It's literally everywhere. It's in the ocean, it's in the water, it's in the air. So the question is, how does it impact us as human beings, and how does it affect our lives? By the way, I do want to point out here that one of my favorite books here in this list is the first one that says "I Contain Multitudes" by Ed Yong. It's a wonderful journey through how these microbes impact animals around us, and in turn how they impact our lives. And also the second book is quite interesting as well. A lot of the others I have not really read them, but they are definitely on the list if you were to go and search for them. Now, instead of telling you about those books, let me start at the beginning and tell you a little bit about what microbiomes are. As I said before, microbiomes, at least for our discussion today, is simply going to be a community of bacteria that lives in one particular environment. And a Scientific American article by Jennifer Ackerman back in 2012 called this the inner ecosystem, saying that within our bodies we have an entire ecosystem going on. And what she pointed out and what she summarized for us was some of the knowledge that we had prior to this. And what we know now, what we knew even then in 2012, was that there are probably more bacterial bacterial cells in our body than our own cells, probably even 10 times more. So what this means is that number-wise, the bacteria and the microbes have a huge presence inside our body. What is even more shocking is that of course, each of these bacteria have probably a few hundred to a few thousand genes. If you look at all the genes that they have and all the genes that they express, it turns out that they probably express 500 to 1,000 times more number of genes than our own body, which is quite shocking. Which means that the microbes in our body have way more activity going on than our own cells to some extent, if you look at just sheer numbers. Now, in order to appreciate these numbers, these mind-boggling numbers, let me show you the next picture. What you see here on the right is a cross-section of a mouse gut, mouse intestine. And what you see, if you look at that picture, the right-hand bottom shows a bunch of blue patches. Each of those is a cell of the mouse tissue, the tissue of the intestine. And as you can see, they're fairly large. And then you see this green band. The green band turns out to be the mucus layer that separates the inside of the intestine from the cells of the intestine. And then the colorful stuff that you see on the top left are essentially, most of it is microbes. They're mostly bacteria. And those are much smaller, as you can see. And this picture should, you should connect this back to the previous slide where I said, hey, the bacterial cells in our body are maybe 10 times more than our own. A picture like this should convince you that it's not surprising. This is very plausible. In fact, if you look at the inside of the gut, especially the large intestine, much of it is just mostly bacteria and the food that is being digested, that is being processed and sent out.
So Giri, a question here is that the fact that there's a lot more of this does it mean more scary, or is it what is
Well,
that supposed to
that's
communicate?
absolutely. So that's my next slide. Perfect question. So what this means is this is part of normal human beings, healthy human beings, and this is what we have right from the time that we are infants. So this picture, the next slide, shows you all the places where microbes are fairly abundant. By the way, in the gut is where you have the most bacteria, no question about it. But they're also there in the nostril, in the oral cavity. There's a ton of them. Even a couple of decades back, we thought the lungs were sterile, that there were no bacteria. It turns out not true. There's quite a bit of bacteria there. Stomach, of course, it's all anything part of the alimentary canal, the digestive system. It's full of microbes. And of course, colon, that's the intestinal part. And then even in the sex organs as well as on the skin, anything that's out connected to the outside environment, it's full of microbes. Now, they all have a role to play. They all have, all the things that you see here on the right, these are all the good things that they do. So in the nostril, for instance, they produce mucus. They also produce some antimicrobial chemicals that keep the harmful pathogens away. Then in the mouth, they assist with the digestion. So throughout the digestive system, throughout the alimentary canal, these microbes are there to help us with our digestion. And that's something that's quite important. And then we'll have a chance to talk. Go ahead, Deep.
Yeah, not all are bad things. That's sort of what it is.
Very few are bad things, and that's what we are going to come to.
Okay.
And the next few slides will show you a bit of the good, the bad, and the ugly, as I call it. So let me start with the good. I've already told you a few of the good things, but here is a more detailed example of a good bacteria. There's a bacteria called B. fragilis. These produce something called polysaccharide A. Again, we don't need to know what they are. They're small. They're chemicals, sugars. These PSAs are picked up by dendritic cells, and dendritic cells are part of our body. They take and present it to something called the undifferentiated T cells. Again, these are technical terms, but most of you probably have heard of T cells, and T cells are part of our immune system. So the immune system, these undifferentiated T cells then become regulatory T cells, and they go and regulate inflammatory T cells. And the bottom line is the punchline, and that says they help reduce inflammation. So what's happening here is that the bacteria is somehow producing things that trigger our immune system. And in fact, this is a role that they play right from the point that we are infants, where we have very nascent immune system, where we have an undeveloped immune system. It's the bacteria that trains our body to become better immune. So that's the good one. Let me tell you about something where it plays a bad role. If we eat food with something called lecithin, I'm not an expert on some of these terms, but feel free to ask. And by the way, a lot of my work was done with one of the previous speakers, Dr. Kalaimati, and she's also on the line. And if you ask questions on the chat, there's a good chance that that you will have the answer even before I get to it. But feel free to discuss things on the chat. So if we eat food with lecithin, it turns out that the intestinal enzymes can convert them to something called choline. These choline go and fuel the bacteria, which in turn produces something called trimethylamine, which in turn becomes oxidized to something called TMAO. Well, these TMAO, after all the technical jargon, this TMAO assists in accumulation of cholesterol and foam cells in the arteries. So what does that mean? Well, we know that this can affect things like heart disease and stroke and other diseases that we are somewhat more familiar with.
So
So
the
again,
one question here, Giri, is that in the sense of the bad, of course, that,
yeah,
how much of what needed to make it a quote unquote a bad impact in terms of cholesterol? Is there a
right.
sense to that, or sense
There
to?
is. That's great. So one of the things that we know is that there is no, it doesn't necessarily, just because bacteria is known to be involved in something like this doesn't mean it's bad. And just because something does good things for us doesn't mean it's a good bacteria. It turns out that they can switch roles, and they can switch
Mhm.
roles fairly quickly.
Mhm.
So good guys can acquire some genes or simply go to a different state and become harmful for the host. On the other hand, uh, bacteria with some bad capabilities don't necessarily turn these on. They're just part of the ecosystem, and life can go on. So there is nothing fixed about when it turns on or off. A lot of this understanding is still somewhat early. Uh, People understand the pathways in which they are developed, but they don't necessarily know the triggers. And I will give you some more examples of things where we know the process, but we don't understand the triggers.
Okay.
And this is where the good and the bad are never very clear. Uh, it's a symbiotic relationship that we have with the microbes, and life goes on. Uh, and here is another one that I call the ugly. This is a picture of a healthy tooth. Of course, again, you shouldn't be surprised that there are bacteria on the surface of the tooth. And if they are good colonizers, if they are there, if these are some of the good guys, for instance, something called Streptococcus sanguinis and Streptococcus gordoni, couple of bacteria, then life is good. Everything goes well. If you decide to turn to a high sugar diet, or if you also have poor oral hygiene, or if you have other factors going on, then you might encourage the growth of a different kind of Streptococcus called Streptococcus mutans, or there are also some other bacteria called Lactobacillus and Veillonella. These guys are acid tolerant and acid producing, which means that they can produce some acids to erode away your tooth enamel, cause tooth decay, cause cavities, and they're generally called dental caries. So, what it also means is that it also depends on what kind of food we eat, what kind of environment we provide to the bacteria, that that might encourage either one kind or the other to colonize, to grow, to become dominant, and to play a bigger role. So life is a balance, even within our body, and much of that balance is also controlled by what we eat and what our own environment is. And so as long as we can keep that balance, life is good. What we also know is that almost each of these environments, here's one more example, which I really like. Uh, uh, Serotonin, by the way, is called the happy hormone. It's when serotonin is produced, supposedly we are happy. But serotonin is something that's important for carrying signals from one part of the brain to another. So for a lot of the nerve signals to take place, it turns out to be important. Serotonin impacts cognition, nervous system. It affects sleep. It affects mood. So serotonin is something very, very important for us. Well, shocking thing is that 90% of the serotonin is produced in the gut. It's produced in the produced far away from the brain, far away from where for a lot of it is needed. And these are induced by chemicals that are released by the bacteria in the gut. So what this means is that the bacteria in our gut have a say in how happy we feel, what our moods are, how well we sleep, how smart we feel for the day, how focused we can be with our work. So all sorts of things. And this has been called the gut-brain axis, and is now becoming better and better understood in all of this. So what it's bringing together is that the bacteria in our gut have a role in almost everything that we do. Now, I don't want to exaggerate it by saying they have complete control over us. That's absolutely not true. But what is true is that they have the ability to regulate some of these things. They have the ability to regulate our moods, to regulate our sleep, but also to regulate heart disease proclivity, and so on and so forth. So it's a fairly complex relationship, and we need to understand this. And it also motivates why we want to understand these microbes. Now, the other thing that's important is that it's not just one bacteria that's doing something. Often it's a community.
Ask a question?
Of course.
Yeah. Serotonin is known to be produced in the daytime when we spend time in the sun.
Yes.
Any relation between the sun and the microbiomes? It's very interesting to know that microbiomes have the capability to produce serotonin. So my question is the sun and the microbiomes.
I don't know the answer to that question. So I can look it up and respond to you separately, but thank you for the question.
I appreciate that.
Sure. So it turns out that there are many places in our body where these bacteria exist. I already told you that. But they're diverse. It's not just one kind that's here and one kind that's there. There is a diverse community in each of these sites. What you find in the mouth is quite different from what you find on the skin or in the gut. And this picture roughly tells you that even though it's one body, we have multiple ecosystems going on, and they are very different. They behave very differently, and they have a different role to play. This paper from 2012.
Let me quickly ask a question.
Sure.
There's a question here: how do we keep our skin microbiome healthy? Is that something you can comment on?
So some of these are biological questions that I may not have good answers for.
Okay.
That one I am not. So one of the things that people need to do is to keep it clean. But beyond that, the question really is what kind of microbes are good for the skin. I don't have good answers for them. Much of this is also, as I said, fairly new territory. There are now new startups that are looking at what kind of probiotics, what kind of things that you can put on your skin that will help the growth of good microbiomes that in turn can help you. But much of that research is still new, and really it highlights the point that we know very little about this domain, and it's also very context dependent. So it really says that we need to understand this within our own context, within, for instance, the Indian context, or the things that we are exposed to. Also, some of it is experimentation. Just because it works on somebody else doesn't mean it's going to work on you, because you have your own unique microbiome. So a lot of the answers are not likely to be easily generalizable. And this is where even the concept of personalized medicine can come in, where you really want to figure out those answers yourself. But there are broad suggestions. There is a lot of things that are already known. And wherever I know a little bit, I would be happy to share them with you. So what this slide shows us is something quite surprising. It tells us that the community that lives in our mouth is very different from the one that lives in our in the gut or on the skin or in the sex organs. In fact, they are very readily distinguishable. And I know the figure is slightly hard to read, but if you see, you'll see that the first one is this first column is something that tells you about the nostril. Then you have the inside of the mouth. Then this is on the tooth. This is under the tongue. This is in the lower intestine. And then finally, this is the vaginal region. And just by looking at the colors, it should tell you that the community is very, very different. But what is a lot more interesting is that even though the community might look different, their functions are, at least within that particular site, are really quite same. So it's possible that you might have more of this bacteria as opposed to that bacteria, but in the end, they're there to play a certain role, and that functional role is somewhat fixed, and that's somewhat interesting to know about this. So diversity, it turns out, is very, very important. If you have a microbiome in your gut that's dominated by one bacteria, then there's definitely trouble for you. So that diversity is needed because they all are working together and playing different roles, but towards a common cause. A good perspective to use is that the bacteria also want to thrive. They don't necessarily want to harm you in some sense. So overall, for their own survival, it is good that their host also thrives. So in a lot of ways, they are not necessarily there to harm you. It's a symbiotic relationship. They want their species to survive and to thrive. And so in some sense, there's a benefit to both sides. And that's the way to think about them. Occasionally, when things go wrong, they do turn pathogenic. And at that time, there are our body itself has some internal systems to correct this. But of course, as we know, diseases are caused, and it needs external intervention to bring back that balance. Well, it's not just our body. I also want to give you an example of diversity in the environment. Most of you who have seen this cartoon called Finding Nemo are familiar with this picture of Nemo and Dory. What is important there from that film, if you remember that movie, by the way, fun movie, you should go watch it, is that these two, Nemo and Dory, live on a reef, and they're surrounded by rays and sharks and dolphins and octopus and seahorses and all these wonderful creatures there. They have friends, it seems. And in the backdrop, what is important is that in this picture itself, in the backdrop, you see lots of corals. And what we know now is that if you have a healthy coral reef, then you should expect the entire array of fishes and organisms, sea life, that must be present. In fact, it's almost natural. It's going to happen. So a healthy ocean environment, there also means that you get a healthy diversity of life. Where in places where the coral is sick and not doing so well, it turns out that the sharks go away. Some of the larger organisms go away, and it starts to impact almost the entire ecosystem. So it's quite shocking, and some of that is caused either by pollution, some of that is caused by warming of the ocean waters. So all the carbon dioxide and carbon monoxide we produce by driving from place A to place B, well, all that is warming up the ocean waters, and that in turn is the coral itself has a symbiotic relationship with the microbes that are living inside them, inside their own cells. And when the ocean water warms up, it releases those microbes, and that symbiosis is now broken. Some of the nutrients that the microbes were providing it are gone, and slowly the coral reef starts to fade and die. Even pollution, when a ship sinks in the ocean, it's not completely harmless. It turns out that it harms the environment around there. In fact, people have found patches of areas where coral just dies, gone, where you have some disturbance of this kind. So it's shocking that the same concepts that we apply to inside of our gut is also true in the environment. So we really ought to think more expansively about these communities and these diversity of microbes, because even though they might be, you might think of them as the lowest form of life, they impact all the way to the largest organisms that might be alive on this planet. Then it's also important to understand how microbiomes are present in the built environment around us, on our desktops, in the buildings, in the ventilation systems, in the cars that we drive, in the gyms that we go and exercise in, restaurants, schools. Everywhere there are communities of bacteria. What is a healthy community? And how should we build our ventilation systems? Are there good building materials we can use that will somehow help? These are all areas, questions that people are investigating. I'm not an expert on it, but it's useful for us to understand that these are the kinds of questions we want to ask in order to put all of this in context. And then, of course, we ask all these questions because we want to understand the world around us, and we want to be able to control the world around us so that our lives are somehow bettered. So all of these are questions to ask about the environment that we have around us. I'm going to now tell you about some large microbiome projects that humanity has gotten involved in. The first one is an important one called the Human Microbiome Project. This was started more than 10 years ago. And the goal of this project was to understand what a healthy human microbiome is. What should a healthy person's gut microbiome look like? What should the microbiome in the nostril look like for a healthy person? So that was their goal. This was funded by NIH. It was a very large project involving thousands of people across the globe. And some of the work that was done as part of that project was reported back in 2012, and the work still continues. The HMP, or the Human Microbiome Project, led to something called the Integrative Human Microbiome Project, which asked broader questions and started to look at some diseases. The obvious disease to look at was anything related to the digestive system. So things like irritable bowel syndrome, things like ulcerative colitis, things like Crohn's disease. These were things that they started to focus on. They also focused on more systemic diseases like diabetes, and tried to understand how the microbes in the gut and in other places in our body might be impacting these diseases. And so the IHMP is another very large project that has generated a ton of data. People are analyzing this and trying to come up with conclusions as to how the microbiome might be playing a role there. Then an even bigger project is the Environmental Microbiome Project. This, of course, the goal is, let's try and understand the world around us, the environment where we live in. What should a healthy microbiome for the ocean look like? What should the healthy environment for the canal or the well or the other water bodies should look like, or the soil or the farmland? what these microbiomes should look like. So this is a large-scale project. It's not as focused as the HMP, where we wanted to understand the human body. Here it's much more expansive, and we want to understand how it starts to play a role. Then there is something called the Global Oceanic Survey. This again was started quite a few years ago, and in fact, this became famous because of a man called Craig Vintner, who went, got himself a boat, got himself some scientists, and made a trip along on the ocean, and ended up sampling ocean waters in all these places to understand what kind of new microbes we can find. And what are they doing there? Things of that sort. And so some of that survey results are also available. And then there have been other projects that are quite interesting, and I'll tell you a little bit more about the American Gut Project and the Human Food Project. Here, of course, they're related more to the human bodies. The American Gut Project simply asked the question, what does the average American gut look like? How is it connected to the food they eat? So this project is, as far as I know, is managed by a nonprofit organization that solicits people to send their samples and send essentially stool samples, and then they analyze them and use these large numbers of samples to form a picture of what the American gut looks like and how it's affected by food and diet and other things that may be relevant. Back a few years ago, they had 5,000 samples. I wouldn't be surprised if by now they have over 50,000 samples that they've analyzed. I don't have the details on this at this point. But of course, it helps them to ask questions like, what is if somebody is eating healthy, what does their microbiome look like versus an omnivore, somebody who eats meat, somebody who eats gluten-free diet, somebody who eats low-fat diet or a paleo diet, things of that sort. So it helped them to ask much wider questions. It's also important to understand that microbiomes it's not a constant. It's not like the community stays the same throughout our lives. They are completely dynamic. In fact, they change even from morning to afternoon to evening. They are impacted by the pets we have. They are impacted by the exercise we do, by the diet, of course, because as we eat things, we also consume new microbes which enter our body. They are affected by sleep, or they also affect the sleep themselves, but they are also affected by sleep. Then, of course, the circadian rhythm, the morning, afternoon, evening cycles. They're tremendously impacted by antibiotics and other treatments and therapies. The picture on the left shows that when inside the body lives in a sterile environment, but as the baby is born, as the infant is born, as it comes through the birth canal, it starts to get exposed to the first microbes. And that's the first microbiome that we encounter. And it becomes part of our own gut and our own body and becomes part of our ecosystem. It takes a couple of years for the internal microbiome of the infant to stabilize, to become mature, and to start functioning in a reasonable way. During those early years, there are lots of dramatic changes to the microbiome, and people have now tracked this and have an understanding of how it changes. There is much of this they also understand now. If the baby is born through the birth canal, their microbiomes are different from babies that are born through C-sections because they are not exposed to the birth canal and the microbes there. If they are born through the C-section, they get exposed to some of the skin bacteria of the mother. And so they're born with a slightly different set of microbes to start with, but in a couple of years, things do normalize and things do stabilize. But there are some differences. So very interesting how some of the early development of the microbiome happens. But over time, things do tend to stabilize. About pets, it's quite interesting that households with pets do engender a slightly different microbiome for the family members. They're exposed to a slightly different set of microbes, a lot of it coming from the pet. But that's not necessarily good or bad. In fact, it turns out some of the research suggests that allergy-related disorders are fewer in households with pets. Again, that's what the data are suggesting. But the complete picture is still relatively unclear. So what this also suggests is, going back to this point about antibiotics, when we take a dose of antibiotics are typically meant for looking at pathogenic bacteria.
40 minutes.
Okay. And these pathogenic bacteria are what we want to get rid of. But in the process, we also kill a lot of good bacteria. So antibiotics are a huge problem. And there is an estimate. It has been suggested that we have lost a lot of good microbes along the way, and many have become simply extinct and have disappeared from our bodies. And that this is something that we should be worried about. It's happening under our noses. We don't really realize it, but it's happening, and that's a scary thought. I'm going to skip this. I know my time is running out, but so let me talk a little bit about the computational side of things. So the question is, how do we study microbiomes? And if I were to, I showed you this picture on the left. If I were to take a little sample from there, how do I study this? Well, I could potentially put it under the microscope, but really looking at it under a microscope is not going to tell you anything. So it turns out that the best way to do this is to extract the DNA from the sample and then take the DNA and sequence it. By sequence, I mean you go and figure out the individual nucleotides in that DNA. Now, since you're taking a sample, there are going to be not only hundreds and millions of bacteria, there are also going to be thousands of different kinds of bacteria. And that makes the problem really challenging, really interesting. So that's where some of the bioinformatics comes in. That's where my role comes in. This is the starting point for much of our work. But really, it's the biology that may be interesting to people. That's why I spent a lot of time kind of setting up the stage. So now I'm going to quickly tell you about some of the computational side. So please bear with me. There are many ways to study the DNA. You can look at just a part of the DNA this approach is called the marker gene approach. Or you can just say, forget it, I'll just look at the whole, it's called the metagenome because we are not looking at the genome of one individual. We're looking at the genomes of an entire sample of microbes. And so that approach helps us to look at this. I'm not going to go through all of this. It turns out that marker gene gives us a small picture, whereas looking at the entire metagenome gives us a detailed picture. I call one as the impressionist view and the other as the realist view. So one is a little more refined and a little more detailed than the other, but that doesn't mean the first one doesn't have value.
So is it more like than micro level and macro level of information even within?
Yes. So the marker, sort of, yeah. Again, I'm simplifying this a little bit.
Right.
Marker gene will get you to the genus level. It won't get you to the species and strain level. But it'll still tell you have a lot of streptococcus or staphylococcus in this community and how much of it there is. So yeah, that's why it gives you an impression of what is there, whereas the other one will give you very fine detail. then you can also study these samples from what is called as a multi-omics viewpoint, which means not only do we try and figure out what microbes there are, we can also ask what kind of genes do they carry and what kind of genes are being expressed, are being turned on. Then you can also ask what kind of chemicals they're releasing. And this leads us to something called metabolomics. You can also look at what kind of resistance genes they have, what kind of antibiotics they're going to be resistant to. Or you can look at the host and say, hey, what kind of mutations there are in the human host and what kind of activities of the microbes are likely to affect them. So we can study these data sets from very different viewpoints, or what we've been doing is to try and integrate all of this and to do integrative studies, which are, of course, a lot more challenging. But again, I'm trying to give you a sense of how the computational approach for some of these questions are. And there are many other refinements of the methods that I can get into, but I'm going to skip all that.
So Giri, a quick question here. When you talk about the data set, how big or how large they are,
good.
Give a context, I guess, would be.
Sure. Very good question. So when you talk about different kinds of microbes, there are probably a few thousand different types of microbes that may be present in a sample. If you talk about genes, there are probably tens of thousands to 100,000 different genes that are turned on. If you talk about chemicals, there are probably also in the range of 5 to 10,000 different chemicals that are produced by a small sample of microbes. In terms of resistance genes, maybe in the order of hundreds. If you talk about mutations, you're probably talking in the range of 1 million or so.
So what is the terabytes?
So in terms of numbers, they may not be enormous, but in terms of the complexity, it starts to get very, very large. Yeah. So terabytes wise, it may not be very big. In fact, it may be even in the order of gigabytes or less.
But it gets terabytes when you have multiple sampling.
Correct.
No, even with all that, it probably is relatively small as compared to some of the big data processing. So if you look at astronomy or things like this, it's relatively small, but it's once you start to analyze it and produce the metadata, it starts to get it's quite in the range of big data processing. No question about it.
Right. Okay.
So that's where some of these things are true. From my own group, we have been involved in many different aspects of this work. We've looked at compositional studies that says what is present there. We've looked at correlational studies. We've tried to figure out which pairs of things or groups of things of these microbes you will find together, meaning which ones may be interacting with each other. We've done multi-omic studies. We've done longitudinal studies, which looks at things as they change over time. We've looked at causality studies, trying to understand what might be causing what. Then from a computational viewpoint, we've looked at high performance computing, we've looked at visualization, software pipelines. We've also done some laboratory validations. Much of our work, we can predict things until we are blue in our face, but unless they're validated and verified, they're not of much value. So we worked with Kalai's lab, with Dr. Mati's lab, and tried to validate some of our predictions. And that work actually is quite interesting. It just recently was published and shows a way of validating some of the complex relationships that we are modeling and predicting. And then we also applied.
Giri, there where your computational work can help is that basically it might help to kind of reduce the setup information in the lab.
Narrow down the possibilities. Absolutely. Yeah.
That saves a lot of time down the road, because
absolutely,
if you look at traditional biology, that was not possible before.
That's correct.
But when you bring in the bioinformatics component to it, that's kind of reducing the number of things to look at, basically.
So of the thousands of microbes and the thousands and thousands of chemicals and so on, we can narrow this down to a small group and say, this is probably what is interesting, what is dominant, what might be separating this group from that group. And then we can go and validate them in the lab. And in fact, the validations checked whether certain chemical was helping certain bacteria to grow, things of this sort. So that's how we isolate the prediction and go and validate them. And we've also been applying machine learning in recent times, trying to figure out if we could predict some of the changes that might happen. So I know my time is slowly coming to an end. So very, very quickly, we've looked at the lung microbiome of smokers. If there are smokers around here, we looked at active smokers and non-smokers, but we also looked at former smokers. And the good news is that if you give up smoking, there is a tendency for your lung microbiome to get back towards a non-smoker's microbiome. And in fact, we find this difference quite interesting. Although it never becomes completely healthy, it does tend towards one direction. So the findings are quite interesting.
Very interesting, this one, Giri. Quick question here is that based on the information, can you tell that that if you stop smoking after X number of years, you are closer to being normal again? Is there something that this analysis actually through the computational process can give you?
We don't have, those would be good questions to ask. For that, right, we will need hundreds and hundreds of samples.
That's what I was getting to. The problem gets very difficult in terms of time.
Problem becomes difficult, but we do have people. We do have samples that we could try to get at.
Okay.
Of course, we would have to go to NSF for money for this, but it needs a larger a larger experimental setup. But it's definitely possible. This is why it's so interesting that
it's a very fascinating.
in a lot of ways, data is our bottleneck.
Right, data is our bottleneck. Right.
If we had the data, we have the ability to analyze them. We have the ability to clarify the picture more and more and more. Mm-hmm. Collecting the data and paying for that infrastructure is the bigger challenge at this point. In fact, that's true about all of medicine and health currently, is what I would say. So the lung microbiome is quite interesting. We've also done work on the vaginal microbiome. We've done some work on the gut microbiome of people with ADHD and other neurological disorders. We've done a second piece of work on the lung microbiome for people with something called alpha-1 antitrypsin deficiency. And what else? We have also looked at infant gut microbiome and much more. Now, what I wanted to say here very quickly is that microbiomes are complex because the microbes interact with each other. They are not just in isolation. They communicate with each other using various signaling molecules. They cooperate by sharing resources. They cooperate sometimes to hunt together, to kill. They compete for nutrients. They show generosity by releasing lots of good things for everybody else, including the host. And they kill each other by releasing toxins and other nasty compounds. So this should remind you of a social network, which is what we do to each other on a social network. We communicate, we are friendly, we cooperate, but we also take each other down, we hurt each other, and we kill each other. So nothing new there. Here's a great picture of a bacteria that works together, forms swarms, and starts to prey on other bacteria. And in fact, those ripples that you see are these bacteria forming groups and heading towards where the food is. And the food is the other bacteria. So they do work together. They do things, but they also hurt each other, and also to some extent hurt the host in certain times. We've done some work on trying to understand this social network by looking at what things occur together. Without getting into details, I just want to paint a picture of some of the possibilities. Then once we built this network of relationships, we've started to study the network itself and try to see what might be important there and what might be telling us a more interesting story. Then we've looked at how to look at longitudinal data and understand some of these relationships. So that's another work that was published fairly recently. Then we've studied infant gut data sets to try and see what might be causative for what else, what might be telling us from the time sequence. And this is work that was published early this year. I mentioned the laboratory validations. This shows some of that work with Kalai's lab. We've also done some work on how to do much of this work on the cloud. And garnering resources that are readily available, that are cheap, without every university getting fancy resources. Any small institution anywhere in the world can now use these cloud resources. For the price of a Starbucks coffee, you can analyze one small data set, which is actually quite nice. We've also worked on visualizing these communities, which is, this is interesting work that my recent PhD student did. It helps us to look at entire communities very quickly, look at differences, look at how it changes over time, look at even, we can even make videos of how the community changes over time, things of that sort. We've also looked at causality, trying to understand what causes what other things, and that's work of another PhD student, Mushfiq. So I should try and wrap up. I know my time is ending. So let me quickly talk about what are possible projects for the Indian context. This is not just something that bunch of scientists are working. This is not something that's far away for you. These are things that almost any institution can do. Anyone with interest, either in the biology side or on the computational side, can do. Really, we should be thinking about what is a healthy Indian microbiome. We can't be satisfied with what is published already, because they are definitely biased towards a different group. We should be understanding what is relevant for our context. And that also means looking at environmental microbiomes, looking at our canals, lakes, oceans, looking at plants, animals, looking at the soil, looking at what might be important for the farmers, and also the man-made spaces. So a lot of possibilities exist, and it's really the young people here in the audience to carry forward some of this, to convince your politicians and your entities to fund projects like this that will help us in our own context for the future, and not just to rely on research that's been done elsewhere. Many of these questions can be replicated and asked very, very pointedly in the Indian context. There are many diseases, there are many things that are much more prevalent in India. And these are things that really we ought to think about from the context of these microbiomes. With that, I'm going to, these are pictures of people I've collaborated with over the years. It's not even complete, but it's just some of the people that I worked with. And I am going to stop and see if there are questions that I could answer. Sorry, Deep, that was, I went great. Probably a minute over time.
No, it's good. Giri, I'm going to start with a few questions I have, and then I will open the floor for anybody who wants to ask from the audience. You may unmute yourself and ask your question, but let me first ask him a couple of questions. So when you are talking about this sort of an interdisciplinary nature of a problem, you need to have, background a bit in biology, obviously, and a bit in computer science. How do we think is that, what does it help to be able to train or learn, or at least get your foot in the door for younger people? Can you comment on that?
Great question. Look, many of my PhD students started with zero background in biology.
Right.
They were trained in computer science and were willing to go and learn. And I myself, I can say that I have never taken a biology class after my 12th standard. Everything that I learned on my own. I learned on my own, and I learned by talking to people, and I learned from Kalai and from other people that I interact with. So this is possible. The world has become much more interdisciplinary,
right.
And you have to take your expertise and say, how can I use this to share with others?
No, what partly I was getting to, Giri, is that when we went to school,
what expertise?
No, when we went to school, it was very streamlined that you go this or that direction.
Yes.
But going to our point about interdisciplinary, my general comment to younger people is that take as much diverse things as possible.
Absolutely.
Even though you might have an interest in computer science, but you have some interest in biology. And then also remember, even with the biology and computer science, there's a social aspect to it, like the one you talk about, Indian gut for that matter, right? So, a little bit of a, sociological background and stuff like that also very, very important. So I know our educational system do not help you train that way, but it's important to kind of keep that in mind. And so that was my first thing. I mean, but I was trying to get at the very early.
No, I understand.
Find collaborator later, that we know, as Kalai said.
Well,
but the problem is that at the early stage to realize that, I can actually do something at this point to at least get some knowledge to that, okay, this is the direction I want to go. So
agreed. But also it is important to cultivate your own ability to say, hey, I want to learn about this. Or if you're a biologist, learn how to become quantitative about your understanding of the field. Mm-hmm. If you're a computational person, learn to think about things outside your domain and think about how you want to apply this.
Correct.
By the way, things like statistics and basic computing and basic quantitative understanding is important for everyone.
For everybody.
Is what I would
Statistics
say.
is so important now. One person made a very interesting comment here. A few years ago, I did a course in Coursera. That's a very important point. That nowadays,
you have access.
You can have access to something online. Yeah. You can go to Coursera or Khan Academy or Udemy. Yeah. These are very cheap. But you can learn something on a basic module basis. To learn about it. Besides your core competencies that you build, actually. Yeah. Very good. Okay, and I'm going to ask you another question. How do you pick your PhD students? Because they don't come with a biology background, and how do you convince them that it's an important problem to look at? So,
yeah, I do look for people with some natural quantitative abilities.
Okay.
Because that's not something I can train. That they have to kind of walk in with.
It's like an inherent kind of training.
It's an inherent thing. So really, that's the only thing that I look for. Some 20 years ago, I started a master's degree program where I looked for people with advanced degrees in life sciences and I said I will train them with computer science and turn them into fabulous bioinformatics people. And then a decade or so later, I went the other way around and I looked for computer scientists for whom I said, I will teach you a little biology and turn you into fabulous bioinformaticists. I have to say that the second experiment worked better than the first. And the reason is that quantitative abilities are much harder to teach.
Right.
So I'd say that perhaps that answers your question. Yeah.
No, that's a very good point. Very early on, training your analytical thinking and quantitative mind.
It's very important too.
You do.
And just because you're a biologist doesn't mean you can run away from math. You can run away from quantitative things.
Not anymore.
It'll help you to think. It'll help you to think in a logical manner, and that is that thinking ability is important.
Yeah. Very good, Giri. Let me open it up. We got a few more minutes. Anybody in the audience, participants, they want to unmute and ask a few questions. Please do so.
This is Subhash.
Okay.
Oh, hi.
Can I ask a question?
Hi,
please?
Yes,
please. Go ahead.
Yeah. Thank you for your enlightenment on this subject. Actually, I'm a big fan of microbiome for several years.
My pleasure.
You know, I am a big believer of Ayurveda treatment versus Western.
Yes.
Because that's a prevention, actually. I look at prevention as a cure for various things. So having said that, I'm looking at nitric oxide. It is a super molecule right now that is being produced, scientifically proven when we do Brahmari pranayama, right? So if we have any bad bacteria in our mouth, okay, that will, the nitric oxide will be offset, right? So we have to maintain hygienic mouth. You touch base on that, nose, for example. So having said that, actually, I looked at the toothpaste we use, which is coming from Western countries' culture, actually. If you look at that, the antibacterial activity in there, it kills everything, right? Mouthwash is another one, which is, detrimental for us. So what I did was I made my own toothpaste based on our Indian spices. Wow. It works wonders, right? And also same way, you touch base on the skin, the biome on the skin, right? So we need to protect that also. So we should not be using soaps and various creams that are being produced today. So hopefully I can make my own soap. It's very simple, being a chemistry. By the way, I'm a IIT PhD, electrochemistry in '84.
Very nice.
I'm making my own soap, sodium hydroxide and coconut oil, olive oil. That's all is needed.
Great.
So these are the things one can do, protect our biome. So I'm really interested in communication with you in future. And also you didn't touch base on the food we're supposed to take. I'm a big fan of fermented foods. Fermented foods, I keep experimenting for the last several years, especially 2020 was a good year for me because stayed at home and working pretty much from the kitchen. I experimented with various fermented foods. Somehow Indians, it's not popular in India, the fermented foods. That's one topic actually that's very, very important for us to protect our biome. Thank you very, very much for your, I have some questions, but I will probably, communicate with you offline.
Absolutely. So a couple of points. Probiotics is a huge field. I actually skipped past that slide. Clearly, the food we eat is important. In fact, for those of you who may have heard of this book called Like Water for Chocolate, it's written by a Mexican author. Every chapter of that book starts with a recipe, and the idea behind the book is that you can influence people by what you cook for them, and then the chapter would have something to do with what she claims is the impact of that food. So, people have known this. Our ancient cultures knew how to control our bodies with food and with other things. Some of that needs to have needs to be made more scientific. And that's an effort that I would really like to see promoted more. The knowledge is there. We have amazing ideas, but we need to, in order to establish the work and provide it a firm scientific footing, I would like to see this validated in the lab, so that nobody starts to question this as just a belief system, and will actually find the scientific processes that explain the connections between the good food and the good effects.
Right. I think the scientific process of to establish it is also very important. Anytime you do something. Excellent. Anybody else have any questions?
I have one question.
Okay, yes, go ahead, Raktim.
Hello, Narasimhan sir. It was nice to listen to you. I have a question. Like the American gut microbiome project is mostly like it was based on the American people, and the
Yes.
food is completely different. Like most of the
Yes.
Americans they go to fast food. So do you think that in the Indian context the microbiome will be more diverse than the American microbiomes?
It will be different. I don't know about necessarily diverse. It will. My guess is that it almost surely has to be different. That's why we need to study this.
Right.
We need to understand what's a good balance for the Indian context. We really don't know this. We can't just go with what the conclusions are from a study elsewhere. We have to do this ourselves. And no one else is going to do it. It's young people like you who have to lead the way.
Because I recall seeing one webinar where they have compared the microbiome of the tribal people in the US and the American cities people. So the one in the tribes, they are more diverse because
they're more diverse. That's absolutely
right.
In fact, there are tribes in Africa that have been studied extensively, and there's clear evidence that they are more diverse, that they have microbes that have disappeared from the Western gut. And a lot of it is connected to extensive use of antibiotics and other things. Yeah, you're absolutely right. And in fact, we might be able to find things that are still lingering in the gut of people in India that might be of value, that might be useful. And people now have treatments where they're able to locate these microbes and find ways to bring them back into the system of people. If we can make a toothpaste with some microbe that we find only in the gut of some people, it's entirely possible that it could change our lives.
Thank you.
Very good question, Rockton. Okay, any more questions?
Well, feel free to email me or get in touch with me. I'm happy to answer more questions. And it looks like the chat session has been active. I'm happy to see that.
It has
thanks
been very
to Kalai.
to Professor Kalai for answering a lot of the questions, and we'll also make the chats available afterward, after Giri and Kalai have a chance to look at the questions to get a response. Thank you very much, Giri, again for giving the presentation today, and it was
pleasure.
Great listening to you on this intersection of biology and computer science and the new possibilities really to look into problems for the newer generation. So that's kind of very exciting.